Hi, my name is Tom Fleming. I'm one of the co-founders and the COO of Arctorus. We're a biotech company based in Oxford, focused on the total automation of drug discovery. My talk today is machine learning and automation, a tech-enabled future for drug discovery. I think we can all agree that we desperately need new medicines. 1 in 2 of us here will be diagnosed with cancer at some point in our lives, and whilst we are able to treat other diseases, over time, age-related diseases, degenerative diseases will catch up with us. The drug discovery process itself is extremely time-consuming and extremely costly, taking over 10 years to bring a new drug to market, with costs upward of $2 billion. In the first part, the discovery phase, we're focused on the identification and development of chemical matter that shows a promise for being a potential drug candidate. This is an iterative process of design, make, test, analyze, as we'll come to later, and it involves teams of chemists, teams of biologists, all working to identify and develop the desired properties in a drug. Not only are the costs to develop a new drug staggeringly high, but in fact it's getting more and more expensive over time to develop a new drug and bring it to market. In the computer chip industry, we have Moore's law, which shows the exponential improvement in computer chip power as the industry develops. In pharma, however, we have the inverse law, ERIMS, which shows that the cost to develop a new drug is predicting every so many years. So costs in pharmaceutical research are escalating, driving up the price of drugs and limiting the amount of funding that's available to invest in other research programs. This is an industry with productivity in decline. Perhaps it is no surprise then that the underpinning processes have stagnated. If we look at the labs of the 1960s and compare them to the labs of 2021, whilst the microscopes on the bench may be far more sophisticated, the setup of every experiment still requires humans to transfer small volumes of liquid back and forth on the bench in a repetitive, menial, and extremely time-consuming way. Whereas all other industries have seen leaps forward in technological adoption, automation, digitization, where progress is now exponential. But the labs, progress remains flat. So what is the outcome of this outdated work mode? Well, data quality in biomedical research is often poor, with as much as 89% of landmark oncology publications being deemed irreproducible by independent scientists. That costs the industry a staggering excess of $28 billion per year in wasted research spending. The labs themselves are extremely expensive to establish and extremely expensive to maintain. This is very luxurious, superfluous infrastructure with extremely low utilization rate. Lab work itself, that occupies the time of our most valuable resource, the scientific brain, is extremely laborious, and highly trained scientists will spend the vast majority of each working week sitting at the bench doing repetitive, menial, time-consuming tasks with a lower data quality output compared to a robotic alternative. This leaves very little time for the most important mental activities such as hypothesizing, reading, analyzing data. So what did we do at Arctorus to address these problems? We developed a total automation solution for fundamental biological research that spans biochemistry, cell biology, molecular biology, and biophysics. If it can be pipetted by a human, Arctorus can automate it to a higher standard of precision, consistency, and reproducibility. We operate a single, centralized platform for a diverse range of experimentation, and we've built up a validated library of drug discovery protocols with their corresponding automated analytical pipelines to massively accelerate drug discovery and generate high-quality data. It is fast, cost-effective, reliable, and accessible globally. Our automated platform is an evolution beyond the high-throughput screening laboratories of the past. In pharma, you can find robotic arms, you can find a high-throughput robotic system, but this is capable only of one single experiment, typically a simple experiment, being repeated 1 million times. And the effort to reconfigure that for a second process, a third process, a fourth process, diverse processes is prohibitive. It can't be done. So Arctorus set about building a robotic platform that is capable of a wide range of drug discovery experiments at lower throughput, and it will interleave and coordinate and schedule these simultaneously. This automation concept delivers precision, accuracy, transparency, and full reproducibility, and there's a massive boost to biotechs and pharmaceutical companies worldwide who access that form of data generation. The Arctorus platform digitizes the experimental process, stripping away ambiguity and generating on average 100 times more data per assay. This is a huge boost to drug discovery research. How does this impact on the problems I mentioned in the beginning? In terms of unreliable data, our automation process strips away variability and enforces consistency and guarantees reproducibility. All data is standardized, structured, and enriched with QA, QC, and audit trails. We remove the financial and geographical barriers to accessing the top-end infrastructure, the top-end laboratories. We remove the need to set up a lab. We can think of this as the transformation that took place when AWS took care of hosting centralized server rooms and replaced on-premises computing power, transforming the abilities for companies all around the world to access high-end computing. In terms of the utilization of our valuable resources, the scientific brain, instead of sitting at the bench all day, scientists are now able to access the Arctorus platform through the cloud and have robots conduct their time-consuming experimental tasks. This puts out a level playing field for the entire world in terms of accessing the cutting-edge laboratory of today. I want to move on to discuss how a fundamental process that repeats in drug discovery is being impacted by machine learning and by automation, and is undergoing a massive overhaul of technological evolution. This is the design, make, test, analyze process, and it iterates throughout drug discovery from the early stage of the first chemical matter that shows promise of being a drug, all the way until the desired properties have been developed. Traditionally, this is the domain of the medicinal chemist for design, the synthetic chemist for make, the lab biologist for test, and the scientific team generally for analysing data. Once your molecules have been selected, synthesized, profiled in the laboratory, the design team will ingest that results and make the next step of design and the next set of hopefully improved molecules until they reach that desired set of properties. Now, Arctorus' automation platform takes care of the test phase. We conduct the experimental processes to create the data packages, to validate, and to feed back into the design cycle. Additionally, our automated analytical pipelines conduct the analysis, ingesting raw data, outputting fully analysed results, massively accelerating the time taken to process data. What impact is machine learning having on the DMTA cycle? Well, it's the design step. Whereas once upon a time the medicinal chemist would have been responsible for setting the next set of structural designs for molecules, instead generative modelling is now taking over this space. This is an advanced form of machine learning which is able to extrapolate into novel chemical space and predict the next best designs to go and synthesize in the lab. And it's important to note that we do need to engage in that real-world feedback loop to actually make the molecules and actually test them in the labs. This puts a crunch onto synthetic chemistry. Traditionally, chemistry would be done by teams of synthetic chemists around the world. But now this is a process that's being automated, but it's lacking. There are many challenges preventing this from being widely adopted. There is the substance variability. Chemicals are often gums or gases, volatile, toxic, explosive or flammable, in stark contrast to the predictable, consistently aqueous nature of biology. There are challenges for inline reaction monitoring, challenges of scale versus throughput, of complexity versus standardization. There are challenges in purification and workup, how to conduct multi-step syntheses without accumulating contaminants and impurities that corrupt late-stage synthetic success. There are also challenges around the validation, the isolation of chemical matter, and its quantitation, especially at low scale. So in terms of trends and outlook, synthesis is rapidly becoming the bottleneck in drug discovery. There's an explosion of machine learning, which is increasingly impacting the design process. Arctorus is automating the tests that analyze, and in concert, we're able to rapidly accelerate through design, make, test, analyze cycles, held up largely only by the speed of chemical synthesis. There's increasing learning amongst this explosion of machine learning companies that the models can only be as good as your data, which puts pressure on the source of data generation. And there's increasingly a realization that robotic data generation is the perfect complement for machine learning based drug discovery. Our data generation is intrinsically precise, consistent, structured, and reproducible. This feeds directly into the richly annotated requirements of machine learning drug discovery, enabling them to make more confident and more effective predictions about potential drugs to test and analyze. Without robotic data generation, people hit somewhat of a glass ceiling in terms of access to high quality data and are dependent solely on publicly available databases such as Chembl. But of course, this is prone to all of the variability and reproducibility issues which we mentioned in the beginning. Automated chemistry itself is somewhat behind automated biology. Whilst there are a handful of solutions and a handful of academic labs investigating this, all current approaches lack the concept of integration and flexibility. There are isolated islands of automation, each taking care of an isolated task. What we need is a tighter coupling between chemistry, biology, and machine learning. We need to consider the entire workflow from design to make, through purify, to test, analyze, and revision of design concept as a whole. We cannot look at these as single steps. This needs to be brought together. Also, the pragmatic need for rapid expansion of chemical matter may also see a renaissance of the combinatorial techniques we've seen in the past. If you were to ask, is automation and machine learning really impacting drug discovery? I think the answer is yes. We've seen companies like Exscientia and in silico medicine already pushing machine learning derived candidates towards the clinic, and both organizations are turning to automation for their data generation requirements. If you ask how is machine learning going to impact drug discovery, this schematic in an abstract way shows the progress from early hit matter through to lead-like properties, that iterative walk through chemical space, finding which structures, which activity relationships lead to that desirable set of properties. We're plagued by false positives, plagued by false negatives, and struggling to develop all of the properties we need. Machine learning, however, combined with robotics, has the potential to make much more creative and effective leaps through chemical space, conducted faster and with the requirement for fewer cycles, massively accelerating that hit to drug candidate process. So my final slide is a call to action. The interface of science and technology is where I believe the next breakthroughs in chemistry will originate. It is not sufficient to build isolated technologies, but rather we need to build integrated platforms. The entire interdisciplinary workflow, from data to chemistry to biology and back again, must be integrated to radically accelerate drug discovery. Thank you for listening, and if you'd like to discuss automated drug discovery and automated chemical synthesis, I'd be delighted to speak with you. Thank you for your time.